I hope by undertaking research focusing in sustainable development, we will be able to create change as part of a continuous improvement process.
Recognize the work of healthcare and life science scientists and organizations to contribute to innovative healthcare solutions.
I hope participants will continue their efforts in showcasing their hard work, find a product, and meet potential partners.
We are growing too fast. We walk fast. We eat fast. Though we have 24 hours in a day, somehow we share the same feelings. Time is passing so fast. We don't have enough time for ourselves until busy is the new norm in our life. But if you can turn back the clock, what good shall you do today? Let's put life back. We need the past to enlighten the present and the future. We need education. Through education, it reconnects you with the purpose of life. Reconnect with family and people. Reconnect with nature and habitat. Reconnect with unity and harmony. Reconnect with spirituality and peace.
اقرأ باسم ربك الذي خلق.
Reconnect with good health and well-being. To create balance, UITM nurtures Buddhi in education and embraced it as values in life. A concept of social relationships, a philosophy and culture of politeness, reasoning and wisdom among Malaysians. UITM upholds Buddhi for sustainable community. Education reconnects you with the universe. Reconnect your world with UITM.
In the quest to search for knowledge and insights, diving deep for ideas to gain prominence, be they the scientists, teachers, accountants, entrepreneurs, engineers, artists, managers, lifesavers, mothers and fathers. Let us be a trusted companion, your preferred high-flying institution, as we achieve excellence holistically. Because we believe in nurturing our future talents, we endure sowing the seeds for a sustainable tomorrow. We harness your capabilities. We take steps and stances to broaden our knowledge and experience. We want to be the champion to uphold mankind's universal values, to encourage a deeper understanding of sustainability. We dare to be different. We instill values. We cherish the diversity, where science and arts complement each other. We go boldly. Set the scene. Steal the show. Think out of the box and set our sights. Walk far and wide. We dedicate ourselves to all our passion, to nurture the courage in all of us. We lead.
UCSI University has come a long way since 1986 as a Praxis University. Today, UCSI is ranked at the top 300 university in the world, rising steadily every year, reaching the rank of 284 in the year 2023. In the same year, UCSI became the only university in the world to receive the QS Recognition for Improvement Award, having improved by the most ranks in average. UCSI programs are ranked among the world's best in music and performing arts, hospitality, engineering, business management, art and design, social sciences, and medicine. As a Praxis University, our students are exposed to a wide learning experience. Thousands of corporate partners offer internship and work exposure to UCSI every year, enabling our students to apply what they learned in the classrooms to be practiced at the workplace. They give the students opportunities to work and learn. This resulted in our graduates attaining 100% employability score in all our programs. One of the hallmarks of being a UCSI student is the research attachment opportunity at world-leading universities at the undergraduate level, enabling them to publicize remarkable papers at the world stage. opportunity to study abroad, whether for a year or two, or for a short term of a few months, prepare our students the global outlook that the world needs today. Preparing as leaders for tomorrow, the students are exposed to real leaders today, from academics and economics, to social concern and topics on sustainability. Our students are set for the challenges today and the future. UCSI offers the largest fields of study in a Malaysian private university, whether in championing healthcare and medicine and pharmacy, impacting research and discoveries in applied sciences, applying skills and intelligence in engineering, influencing communication in social sciences, igniting the passion and creativity in arts and design, setting the global pace in hospitality and tourism, transforming architecture and built environment, celebrating with music and performance, excelling in the changing demand of the business world, or digitizing the world in computing and innovation. UCSI continues to serve with scholarships, producing tens of thousands of practitioners and young leaders from more than 110 nationalities. Beside its current main campus in Kuala Lumpur, and other campuses in Kuching and Spring Hill, UCSI has earmarked its Kuala Lumpur campus to expand as an edu city with 35,000 student capacity with various state-of-the-art facilities. Our students are the real stars of UCSI. They achieve and excel in the national and international stage. Driven with the passion to contribute, they prove their quality, participating in various events and attaining many successes. UCSI enables them to make the mark for themselves, build their own reputation, and create a brand for themselves to face the exciting world ahead of them. Every year, our medical students advance their undergraduate medical research at the Harvard Medical School. Others made the engineering impact at the Imperial College London, at Queensland, Australia, and Tsinghua, China. While some set their pharmaceutical legacy at University of Chicago and British Columbia. These are some of our students who have benefited from our Star Trek to the world's best universities. The world is changing, and they are at the forefront. The needs are evolving, we continuously confront. UCSI, a Praxis University.
Ladies and gentlemen, our next program is a webinar by the National University of Singapore on an academic journey for sustainable development in the pharmaceutical industry. Please enjoy.
And good afternoon. I'm Izzat Sufyan, Assistant Professor from International Islamic University Malaysia, who will be your host for today's international webinar. So as everyone knows, looking at this poster, so our topic for today is an academic journey for sustainable development in the pharmaceutical industry, organized by Malaysian Technology Expo 2022. Again, welcome everyone for attending this event. And today we have a list of our tremendous speakers from National University of Singapore, specifically under the Department of Pharmacy. So without further ado, I would like to invite Professor Christina Chai, Head of the Department of Pharmacy at National University of Singapore, to give some introduction on National University of Singapore Department of Pharmacy. Please welcome.
Hello, everyone. I'm Christina Chai, and I'm the Head of Department of Pharmacy of the National University of Singapore. I would like to welcome you to this webinar, and I would like to give you a very short introduction to the Department of Pharmacy. The first question is, who are we? As you can see over here, we have a very, very long history. And actually, our history started while Singapore was still part of Malaysia, and hence, we have a 117 years of history. In terms of the quality of our research and education, this is measured by the QS ranking. And in terms of the world in 2022 for pharmacy and pharmacology, we rank number 12. So what is our values? Our values is basically that we believe in making a difference. And this is in terms of education, in terms of practice, and in terms of research. Let me talk a little bit about our research. When we when I show you in this slide over here, you can see that the vision statement for our department is to be able to carry out innovative and impactful research that will be able to transform and save lives through new and enhanced knowledge of health, diseases, and drugs. What is truly unique about our department is that it is a one-stop shop. So it is one department with multiple expertise from chemistry, biology, clinical, computational, analytical, and engineering. So the research that we carried out is actually from fundamental to applied research. And this is one of our strengths. We have faculty members from different disciplines, which means that our education will actually be holistic. In terms of our research, as I have outlined, we have multiple themes, and it can range from drug discovery to drug delivery, to biology of disease, to health outcomes research. And they're all bits of a puzzle that is going to fit together. So our research is in fact integrative, which is going to span the whole value chain. So what's our value proposition to you all today? We believe in the promise of education. And specifically what we have today to introduce to you all today is our Masters of Pharmaceutical Science and Technology. It embodies a cross and transdisciplinary program. And to note that we are the only tertiary pharmacy and pharmaceutical science education provider in Singapore. And what we have is our ability to integrate the research with the learning, so that what we teach you is effectively going to be the state of the art. And the importance of pharmaceutical science and technology is really evident. Just in Singapore itself, which is a very small country, you can see there's a whole bunch of pharmaceutical industries and also biotech companies that are in fact in operation. And no doubt that in Malaysia, you will be able to see something that is in fact very, very similar as well. What my colleagues is going to be able to share with you today is in fact a little bit about the program, which is the Masters of Pharmaceutical Science and Technology. And then our three colleagues will share with you their research to give you an idea of the expertise of the department. I would like to urge you all to stay connected with NUS Pharmacy. So if you scan this QR code, it will in fact lead you to our social media pages, okay? Because what's the best way in which you're going to be able to know us is to be able to follow us and to see what we are up to. I thank you all for your time today, and I hope that you enjoy the presentations that will follow. Thank you very much.
Right, thank you to Professor Chai for the introductory of our program of the NUS, mainly the Department of Pharmacy. So next we go with the next session. So the next session we will have a presentation of academic overview of postgraduate education program at NUS. Will be presented by Associate Professor Dr. Gigi Chiu, the Deputy Head of Department of Pharmacy Education in NUS. So without further ado, please welcome Professor Dr. Gigi Chiu to present the program.
A very good day to the audience here. I'm Gigi Chiu, Associate Professor and Deputy Head of Department of Pharmacy at the National University of Singapore. In the next 15 minutes or so, I will be sharing with you our postgraduate education programs and how we nurture future leaders for the pharmaceutical industry, as well as academia with unique and enriching learning experiences. As the only institution offering education in pharmacy and pharmaceutical science for 117 years, we are proud to be Asia's top university based on US World Ranking, and number 12 in the world for pharmacy and pharmacology. We attract the best students and faculty members to our department. Besides our undergraduate education, which include both Bachelor of Pharmacy and Bachelor of Science in Pharmaceutical Science, for our postgraduate degree programs, we offer PhD and master degrees by research, as well as 2 coursework degree programs, the Master of Science in Pharmaceutical Science and Technology, and the Doctor of Pharmacy, which is an advanced degree program for the training of clinical pharmacy practitioners. For this presentation, I focus on PhD and the Master of Science in Pharmaceutical Science and Technology, and how they are aligned with the booming pharmaceutical landscape in Singapore. The pharmaceutical and biomedical sciences and manufacturing is an important sector in Singapore. The map here not only shows the island itself, but also the multiple pharma giants in various industrial park locations, where they have established their manufacturing operations, as well as R&D centers and facilities. All these contributed to a vibrant landscape of the pharmaceutical industry in Singapore, making Singapore an important hub in Asia for pharmaceutical R&D and manufacturing. In particular, the pharmaceutical and biomedical manufacturing forms the 4th pillar of manufacturing economy. And the Singapore government is very supportive of pharmaceutical R&D and commercialization, and has injected a $4 billion budget in 2016 to drive such activities. In 2019, the pharmaceutical and biomedical manufacturing sector has created over 24,000 jobs for the Singapore economy, creating a favorable economic and business environment. So there are a lot of opportunities and excitement in the pharmaceutical and biomedical sectors in Singapore. As for our department, we have a group of committed faculty members and staff who are recruited from top-notch universities and have extensive research expertise in the area of pharmaceutical formulations, development and manufacturing, and drug delivery, with strong collaborations with the pharma industry. So you're welcome to visit our department website for the individual research profiles of our faculty members. In this 21st century, the key driving force in the development of the pharmaceutical industry is sustainability, whereby the advancement of pharmaceutical products is critically governed by innovative design of materials, processing systems, as well as delivery technologies. For the current research of our department, our faculty members leverage on the principles of green chemistry and green engineering to drive the development of innovative nanotechnology-based materials and drug delivery systems to bring about a huge impact in both disease prevention and treatment quality. And these exciting research areas are the focuses of our PhD research program, where our PhD students are mentored closely by our faculty members to advance the frontiers of pharmaceutical research. So here is a quick overview of our PhD program that is marked by curiosity-driven research involving interdisciplinary collaborations with integration of pharmaceutical R&D know-how and innovation. Our PhD program has a duration of about 4 to 5 years and is supported by numerous attractive scholarships that cover full tuition fees, as well as a monthly award for the students. In particular, there is the NUS Industry Relevant PhD Scholarship that fosters the collaboration between the pharma industry and academia, where our PhD students will take on real-world pharmaceutical challenges with their creativity and problem-solving skills. So how to gain admission into our PhD program? You need a relevant bachelor's degree with honors with good standing from a reputable university and a master's degree. In addition, you have demonstrated a strong ability to pursue research in your proposed field of advanced study. For those whose undergraduate language of instruction is not English, you need to provide TOEFL or IELTS results. And starting from August 2023 intake, all PhD applicants must submit GRE scores. In addition to the PhD research program, our department has also started the Master of Science in Pharmaceutical Science and Technology, or MPST in short, in 2008 as a self-funded part-time postgraduate coursework program to provide manpower training for the pharmaceutical industry in Singapore and beyond. The mission of MPST is to nurture industrial leaders who can create positive impact on health of people at the global level with their expertise in pharmaceutical science. And I'm very excited to share with you that starting from August 2022, we have expanded our MPST program to include the full-time option with shorter duration of candidature and open to outstanding students from local and overseas to apply. So what are the key features of our MPST program? This program offers you unique learning experiences where students will have multiple opportunities to interact and network with personnel from the pharma industry. Not only would we invite industry experts as guest lecturers to share their experiences and discuss cases and latest developments in the industry, but at the same time, our full-time students will be doing group projects with part-time students who are mostly working in the pharma industry so as to promote peer learning. We have also taken the blended learning approach, and students are taught in small classes to stimulate collaborative, team-based discussions of real-world pharmaceutical cases. With the presence of multiple pharma giants here in Singapore, what is in the pipeline of our MPST program will be the in-course internship opportunities from the pharma companies to further enrich our students' learning experiences. Comparing to other similar programs on pharmaceutical science and technology from other universities, NUS is the only university in Asia and among the top 50 in QS ranking, with reasonable program fees and cost of living. So now on to some key details of our MPST program. It is a 40 modular credit program, meaning you can graduate from the program when you have earned 40 module credits and with good standing. That is a CAP or grade point average of 3 and above. For each academic year, we have 2 intakes for the MPST program, one in August and one in January. This is to give flexibility to your planning to be admitted into the program. You're welcome to visit our department website for the details of the application period for the respective intakes. The duration of the full-time program is typically 1 year or 2 regular semesters, whereas the part-time program will take typically 2 years to complete. And the program fee for the academic year 2023 to 2024 is shown here on the screen. Within the 40 modular credit program, our MPST students will take 20 modular credits in core modules, which form the foundational and fundamental knowledge important to pharmaceutical science and technology. The remaining 20 modular credits are elective modules to be chosen from a range of modules that cover different skills-relevant topics, specific to the different segments of the pharma value chain. Shown here are some sample study plans to guide you based on your interests and aspirations. For those who are enthusiastic about pharmaceutical R&D, you can do the elective research project and other related modules that focus on discovery and innovation. For those who are interested in product development and manufacture, there are elective modules catered to the technology and processes involved in manufacturing. Last but not least, we also have elective modules that cover the regulation and quality control of pharmaceutical products. And our department will constantly review our offer of elective modules and would mount new ones to meet the fast-changing needs of the pharma industry. So how to gain admission into MPST program? You need a bachelor degree in relevant majors with good standing. Besides pharmacy or pharmaceutical science background, we also welcome students with other relevant majors, such as chemistry, life sciences, chemical engineering, or bioengineering to apply. So feel free to share this information with your friends if they are interested. For students from overseas, you will apply to our full-time program by direct admission, and you will be charged by the program fee. As for the part-time stackable admission route, this is for local Singaporeans and permanent residents, and this route would be charged by the modular course fee. During their postgraduate journeys at NUS, our Center for Future Ready Graduates, or CFG, will organize throughout the year various career events, such as recruitment talks and fairs, as well as services including career planning, resume critique, and interview practices to prepare students to move on to their next stage in life after graduation. In terms of our graduates and their career prospects, so far, we have graduated over 130 MPST students since 2010, and about 250 PhD students in the last 2 decades. Our graduates have either moved on to academia and become professors or lecturers, or secured jobs in the pharma industry and promoted to senior positions in various segments of the pharma value chain, working, say, director of technology transfer, or head of product development and management, or as regulatory consultant in both Singapore and around the globe. And before I end my presentation, I would like to share our beautiful campus with lots of green spaces, as well as Singapore with its reputation of a thriving metropolis and ease of living. So, start your learning journey in this garden city for a jumpstart into the pharmaceutical industry and academia. So join us today and also stay connected with us on various social media platforms. Thank you.
Hello everyone,
So we are going for the Q&A session. So with us today, we have Assistant Professor Yichu herself. We have Dr. Dun Jan Nan and Dr. Chen Wen Chen. We asked to answer all the Q&A sessions. So for those who have questions, you may use the Q&A function button in Zoom, or you might raise your hand if you have any question to ask directly to our speakers today. Hello, Professor Dr. Yichu. How are you?
I'm good, thank you. So how are you?
I'm good. How's the weather in Singapore?
Weather is okay as usual, hot, and okay, today is sunny, we don't have rain.
Yeah, even here in
After
Malaysia
a couple
also,
of days.
it's sunny. It's quite rare as well now in Malaysia we have sunny, because like it has been like raining almost every day.
Yeah, it should be the monsoon season, right?
Yeah, it's the monsoon season is coming soon. Yeah, before we waiting question from the speakers, like I do remember because my last visit just before MCO 2019, bringing my students to Department of Pharmacy there in NUS. And we were so amazed with the whole ecosystem, the facilities, and when we visit the hospital as well. So I'm quite interested on the MSPT program that you just shared with us just now. Like, so the program itself, is it like mainly like they will have connection to industry during the program, or the student, him or herself, need to initiate to have some kind of internship or practical during the program itself?
I would say both. meaning that during the program, in terms of the classes, not just our own faculty members who would actually teach the class, but at the same time, we also invite experts from the industry to serve as guest lecturers, all right, and deliver the classes. So that's one aspect of the NPST program. But at the same time, we also putting up like internship opportunities for our NPST students. And basically, this is for full-time students who are enrolled in the program. Because for part-time students in the NPST program, they are already working, working adults, and they are actually upgrading, upskilling themselves. And so, internship may not be as applicable because otherwise it would be too taxing for them to juggle like a daytime job and studies. But for our full-time students, then we would have internship opportunities for these students.
Okay, that's like interesting because like it's not many universities, even not just, I'm not talking about even Malaysia, like part of the world that offer this kind of industrial appropriate pharmacy program. And then, I mean, knowing that how Singapore, all the technological engineering biopolice, I've been there to biopolice as well, like it's a very good connection and networking to actually involve with industry partners over there.
Yeah.
So for example, like now if Malaysia students do some Malaysian industrial people that are interested to apply for the program, so what will be the prerequisite for them to apply at present?
Okay, so basically from the video presentation just now, I would say the students should have a relevant bachelor degree, meaning that they are coming from a science background, let's say, let's say chemistry or life sciences or biomedical sciences, or some of the engineering program, chemical or medical engineering. So some of the engineering background kind of students will also be relevant, okay, to our NPST program. And in fact, we do have really, these would be the students that are commonly applied to our program and actually studying in our program. Yeah. So of course, I would say it's competitive. So basically, students should have good performance in their bachelor program, okay, to apply to the NPST.
Okay, because I also mentioned about like, is it the GRE also applicable for this or just for PhD program?
GRE is for PhD program. For masters, we do not require GRE. So it is more important for the students to fulfill the English requirements.
But like, for example, like Malaysian, most of Malaysian university, like, I mean, they're delivering the lectures in English, but then of course, like they don't say their degree in English, but the whole ecosystem English. Would that telling them they still need to take the IELTS or TOEFL, or it's not really required for them?
It is not, I would say, okay, it would be safer, okay, to have the IELTS or TOEFL results. Yeah.
Okay, yeah, that means that at least you secure like no question afterwards they want to apply.
Right. Yes.
Okay, so with the current the program that you just shared with us right now, like you did mention like some of them go to academia, some of them go to industrial, some with the corporate as well. Like how exactly, like in terms of the employability after they finish the program towards they got the secure the job?
Okay, in terms of the career prospects, so far, okay, we are running the full-time program for the very first year, all right? So we cannot say for sure, but on the other hand, with Singapore, I mean, in terms of the pharmaceutical industry itself is really, or pharmaceutical/biomedical sector. So it is actually really like booming and doing quite well. So there would be a lot of job opportunities out there. Especially if our students are, let's say, interested in internship, let's say, right? Then that is another opportunity to for our students, to after the internship, consider moving on perhaps with the company itself.
Yeah, because usually like if you build a really great repertoire with the interns company, then usually they will absorb you afterwards to become their permanent employee afterwards, right?
That's right. So even for our part-time students, so it also the NPSD program actually helped them to move on to, for their career progression, to have like a promotion or change to a job scope that is more in line with their aspirations. Yeah. So this is the type of feedback that we gathered from our at least from our part-time, NPSD alumni.
I think it's good because like not many, like you have options. If you're still working, you can go for part-time. If you will go to full-time, you can go for full-time. I think there's options for them actually. So we also have
we
one
provide the
question.
options, that's right.
Yeah, options. I mean, actually for this working, right? Because like
Yeah.
I want to pursue this, but then like there's not many opportunity for that. And the one that you offer is really like very specific program and got options for part-time. I feel like, I mean, if I'm still a student, I would like to apply, but already working, like I don't know, maybe like I can apply after that. I don't know. Okay, we have questions here. So the question is like, hi, how about the PhD program? I'm currently doing my master's. So if I'd like to join for the PhD, I would like to carry my current research as there's a lot of prospects since I'm studying about adaptive drugs. Will there be any like grant, scholarship, or financial assistance being provided by NUS?
Yes, basically, just now during the video presentation, NUS would have a range of scholarships for PhD studies. So not just for, for international students. So that means it is irrespective of the nationality. Yeah, so I guess if my colleagues can share the screen on some of the QR codes that I think I could encourage some of the attendees here to perhaps scan with the QR code to go to our website or to the relevant website that will have the information on some of these scholarships available for PhD studies. Yeah.
Yeah, maybe like the host will be able to share the QR of this. So yeah, I think that's always, I mean, that's quite common question even for me as an academic, like students approaching us like, do you have any financial assistance? I would like to pursue this and this. But then like having like NUS, knowing that the prestigious university in Singapore and also in Asia and the world, like people would like to know, like actually the international students would like to go there, but in terms of financial, maybe their own country have limitation that you provide for the international students to pursue. So I mean, yeah, I mean, like later we will share you the QR.
Okay, so I have put down the link. Okay.
Okay, the link. Yeah.
To the web page. Okay, that is linking to the scholarship information. Basically, it is administered by the NUS Graduate School. Okay, so more information can be found from this particular web link.
Okay, thank you, so Professor Dr. Gicho. So like for those who like to know more about the scholarship, I think not limited to PhD program, but perhaps the other program as well. You can go for the master's program too. You can go to the link being provided in the Q&A box there. You can click under the answer. There's the link. You can just click the link and go straightly to inquire further ahead and to know more of the details of scholarship being offered by NUS there. Okay, do you have any other question from the members of the floor? If not, thank you so much to Professor Cho for the sharing. Like, now I feel like should I continue back my life as students or not to continue my study in Singapore? It seems so interesting to pursue ahead in this because I'm doing drug delivery. I think maybe this is my best opportunity to.
Oh, okay.
All right. After this, yeah. Okay, thank you so much.
So later on, we will have some more presentations that is research focused. Yes.
So all participants available online right now. So we have more presentation after this from all the tremendous research and speakers. So before I inviting them, let me introduce why exactly we choose this topic for today. So like why I think a lot of people talking about sustainability here and there, but why is sustainability being the main topic when we talk about in the pharmaceutical industry? Because like talking about this, like in 21st century, people try to advance the pharmaceutical product, not just in terms of efficacy of the function of the product itself, but how was it the product being sustainability into the material, into the processing, and so forth. So from there, especially we talk about nanotechnology, like it's a big thing for many people how to integrate nanotechnology into the efficacy, into the sustainability that being brought by the United Nations SDG there, like to integrate with that to improve further ahead into the formulation, the technologies, and also we talk about innovative drug deliveries in this case. So that's why with that kind of idea, we choose the topic of like sustainable development in pharmaceutical industry, especially we like to hear from the perspective of the academic journey in this matter. So with us today, we have three speakers, researchers, experts who will share their research with everyone. So for the first one, we're going to have, let me introduce our speaker. For the first talk, we're going to have Associate Professor Dr. Rachel So Professor Rachel actually received her Bachelor of Science degree in Pharmacy with first-class honors from NUS and also PhD in Pharmacy from College of Pharmacy at University of Illinois, Chicago. And after she's done with her doctoral study, she worked as a research scientist in France in drug discovery and also development in proscalar SAS. Later, she joined back her alma mater as in 2006 and now currently hold multiple roles such as Deputy Head of Research, Director of Pharmaceutical Science Program in NUS Pharmacy. Not to forget, she also a registered pharmacist. So not just a researcher, she also a registered pharmacist with the Singapore Pharmacy Abroad. Professor received numerous national grants, national awards, including DAAD German Academy Exchange Fellowship, Banco Santander UCLA W30 Women Leaders Class of 2014, and the Asia University Association Fellowship in 2020. So Professor and her team has quite a numbers of patented work in area of hydrogel and therapeutic peptide delivery. So for today, Professor we're going to deliver her topic, Sustainable Nature-Based Materials for Pharmaceutical Application. Without further ado, let us hear the talk from Professor. Please welcome.
Hello everyone, and I am Professor Rachel E. from Pharmacy at the National University of Singapore. It is indeed my pleasure to be speaking today at the Malaysia Technology Expo 2022. I'm going to share our work on sustainable nature-based materials for pharmaceutical applications, especially in the area of hydrogel formulation. A little bit about myself. I am an alumni of NUS Pharmacy. I received my PhD at the University of Illinois at Chicago in the field of pharmaceutics. Currently, I'm also the Deputy Head of Research and Director of the Pharmaceutical Science Program in NUS Pharmacy. So as we think about sustainability, the topic on circular economy is key for discussion. A circular economy is based on the principles of designing out waste and pollution, keeping products and materials in use, and regenerating natural systems. So this requires us to shift our mindset from use and throw to one where resources can be reused as long as possible. So our goal should be to reduce waste, to recover resources at the end of the product's life, and channel them back into production or use. This will significantly reduce pressure on the environment. The big question for us in the pharma industry really is whether a circular economy is possible. To achieve sustainability, a more holistic approach to overall drug development is needed. So traditionally, we think of sustainability as focus on environmental impact, but the definition now has adopted a wider lens to include both profit and, crucially, people. And when people is concerned, health is what we do. So as a lab, we have embarked on steps to consider research to reflect these values. With our expertise, we think we could do more in considering sustainability in the selection of pharma raw materials. And a few years ago, we partnered with Rocket, a French multinational company specializing in plant-based raw materials, and started a program mining new excipients for hydrogel formulation. Today, I will share with you some of what we have done. So before that, let's start by describing what are hydrogels and why we use them, why are raw materials so limited, and what are the various natural resources that we can tap on to make these hydrogels. So hydrogels are aqueous networks of cross-linked polymers. The internal structures are similar to extracellular matrices found in the body. On the left here, you can see that a polymer powder is being added to a drop of water, which became a hydrogel after cross-linking. Even After dehydration, the hydrogel retains its shape, and the sesame seed is used as a scale. And the pictures below show that you can actually envelop different things in a hydrogel structure, for example, color-coded beads, or even fibroblasts, real cells that show that they are pretty happy in the hydrogels itself. And in a natural environment, our extracellular matrix is actually a hydrogel. But what is special is that these hydrogels do have cell receptors for the cells to form focal adhesion points and attach the matrices. As you can see, the cells being entrapped in the matrices. Synthetic hydrogels do not have that functionality. And there are many examples of hydrogels in the market, such as soft contact lenses, wound dressings, even fillers for cosmetic use. So many polymers found in nature can be used to form hydrogels, but we have yet to fully maximize their potential in the biomedical area. We hope through our research, we can expand this repertoire and advocate the use of natural regenerated sources of materials for health and biomedical applications. So amongst the natural polymers, alginates and gel and gum are the easiest to cross-link without chemical modifications or the need for complex solvent systems. Gel and gum is particularly underappreciated and under-researched because its utility was discovered much later than alginates. There are two forms, the high acyl form, which has acetyl and glycerol bulky groups in the structure. Therefore, due to steroid hindrance, it can only form soft elastic hydrogels, which is not very suitable for biomedical applications. For our study, we work extensively with low acyl gel and gum. As mentioned just now, that the extracellular matrix, which is essentially hydrogel in our body, is bioactive. It has cell attachment receptors. But for a material like gel and gum, it is not bioactive. It does not have all these recognition moieties in its chemical structure. And when we envisioned the use of a hydrogel, we wanted to push the frontier and use it as a scaffold for cell therapy. So this gap led us into thinking about forming an interpenetrating hydrogel, interpenetrating network or IPN network hydrogel between gel and gum and another polymer to confer the gel and gum with cell adhesivity without the need for complicated chemistry. And so we thought we could use collagen because collagen is well established, it's natural, it's found in the body. However, the process of forming an APN between gel and gum and collagen is not so straightforward because gel and gum and collagen have opposing temperature gelation mechanisms. Collagen is known widely to have a heating gelation mechanism, maybe up to 37 degrees. On the other hand, if you see this picture, gel and gum goes through a cooling gelation mechanism. From hydrogels, gel and gum needs to be dispersed as random coils in high temperature, and then the coils will immune to helices when cooled. Weak gels are formed at this stage. To make it a stiffer gel, you need to add cross-linkers such as cations, which will form salt bridges between the coupled slick groups on the polymer backbone. And an interesting fact is that this coil helix transition temperature is concentration dependent. The higher the concentration, the higher the gelation temperature. And so before we describe to you our concentration optimization process to mix the two polymers with opposing gelation mechanisms, there are two other concepts that we need to explain. The first is that cells like mesenchymal stem cells only adhere and spread on a sufficiently stiff hydrogel matrix. And some studies have shown that you need to have at least 1600 Pascal as G prime, which is storage modulus, a measure of gel strength, for cells to nicely attach. And once they attach, they can grow and proliferate. Without that, they cannot. And there's also other studies that show that if you mix gelatin, which is a hydrolyzed from collagen, into a hydrogel network, you could also increase storage modulus by 2 to 3-fold. This information important to us because when we were optimizing the gel and gum and collagen mix, we had run into a little bit of problems, right? Trying to understand what is the desired gel stiffness for cells to grow and to proliferate within. So using rheology, we mix gel and gum precursor solutions, and we can see that there is a crossover. The crossover of the storage modulus and loss modulus happens at this particular temperature, again called the Coy helix temperature, and that there's differences even between 0.4% and 0.5% of gel and gum. There's a difference of 1.7% of this transition, and this has important significance to what we will do next to encapsulate cells in the process. So with the aim of studying the potential of gel and gum hydrogels to deliver cells, we envisioned this scheme, which would have many applications downstream, where we could form a semi-IPN of gel and gum and another polymer such as collagen, or a full IPN with different stiffness. And this hydrogel in turn can be a biologic wound dressing that could contain stem cells or fibroblasts for wound healing or even for burn management. So we next characterized the physical properties of a hydrogel, and as you can see from the picture here, the gel and gum solution and gel and gum collagen mixture, they do not form a gels because you can see using the tube inversion test that they are at the bottom of the tube. Whereas after we added the cross linker, we can actually have them stay at the tip here, showing gel formation. And when we push the hydrogel, the precursors through a syringe, we can retain the shape of the gel as shown. And looking at the microstructure under scanning electron microscopy, we show that the largest pore size could be 150 microns. And this is good because usually stem cells are just about 20 microns in size. And this ensures that our stem cells can actually flow and move within the hydrogel and allow exchange of materials, waste materials, and nutrients within the hydrogel. We wanted to know what is the capacity of hydrogels to absorb water or fluid. This would simulate the ability of hydrogels to take in wound exudate at the wound site. And we find that it could go all the way up to about 160% of our original weight. This is the equilibrium. And over a period of 21 days, our hydrogel hardly had any change in the mass, which means that it is quite stable over time. And this will eventually allow any encapsulated cells to work and exert their biological effect at the site of application. So after encapsulating cells in our hydrogels, we wanted to really show, like we say, that the cells grow and proliferate. So on the left here, we have done a simple MTS assays, which show that cells grown in collagen, which is the gold standard for encapsulating cells and growing them, as compared to our IPN hydrogel, there's no statistical difference, which means that the cells do like our hydrogel. And over here, using a green fluorescence tag phalloidin, which is a protein that binds specifically to cell cytoskeleton protein, you can see over time the cells grow and spread out within the gel itself. Next, we perform what we call 2D scratch assay using the harvested media on human dermal fibroblasts. The migration of this cell line will actually stimulate the wound closure process when the skin is healing from an open wound. The human dermal fibroblasts were exposed to either normal media, stem cell conditioned media, or our hydrogel conditioned media, or serum-free media. The normal media is a positive control because they contain fetal bovine serum, and the growth factors found in FBS will allow the dermal fibroblasts to grow and migrate in a normal fashion, closing the gap. In contrast, in the absence of serum, cells stop growing and migrating, so the serum-free media serve as our negative control. The distance of the gap between the cell ages will image and quantify different timelines, and based on the results, it's apparent that the encapsulated stem cell secreted chemokines in the media, which then promoted the cell migration. And this rate is very similar to the fibroblasts exposed to normal media over here. So therefore, the results prove that our stem cell hydrogels could promote cell migration and hence improve wound closure rates. So how well would our cell-laden hydrogels perform on an animal model? This is a murine third-degree burn wound model. And you can see the same 4 groups of treatment is being used. And the media was exactly what is described in the previous slide. From our data, it is apparent that early wound closure was enhanced by both the stem cell conditioned media, right, as well as the stem cell laden hydrogel. However, this effect was more prominent if live stem cells was present in the hydrogel matrix. We believe that live stem cells serve as cellular factories, continuously producing wound healing factors, whereas loaded wound healing factors were rapidly cleared from the wound bed. Hence, the stem cell conditioned media enhanced wound closure promoting effect was not as sustained beyond 7 days. Visually, scars appear to be less prominent too. The polymorphonuclear lymphocytes, or PMN leukocytes, are cells with multi-lobe nucleus, such as neutrophils and basophils. They are early inflammatory cells recruited to the site of injury. And you can see with our stem cell application on the animal, we can actually have a very strong reduction of these inflammatory cells, proving that our hydrogels were moderating actually the wound inflammation. And visually, we saw there was significantly less scarring. And this is also illustrated when tissue samples were stained with Mason's trichome stains after 21 days. The blue color stains connective tissues, while the pinkish reddish hue stains cell structures. Now, skin injury is only considered to have regenerated when the healed skin exhibits high degree of dermal differentiation. That's why a high degree of dermal differentiation refers to the presence of organized, differentiated types of cells from the layers of skin, giving rise to sweat glands, sebaceous gland, hair follicles. So you can see from our data that it's clear that the stem cells encapsulated hydrogels increase chemotactic factors and differentiation signals to promote a more complex dermal structure and hence dermal regeneration in the full thickness burn wounds that we have used. So at the same time, during our study, we also considered the scalability of production of our hydrogels. And so to align with our sustainability principles, we considered 3D printing as a greener way of production. So at our facilities in NUS, we use an extrusion-based 3D bioprinter known as BioX. BioX has multiple print heads, each with its own temperature control unit, so it does really suit our purpose. And because, as we mentioned, gel and gum and collagen precursors need to be maintained at separate temperatures, so the solutions won't spontaneously form weak gels and clog up the pores of the 3D printer. So using this BioX 3D printer, we wanted to determine if the material can be extruded. So what we did was to do rheological measurement alternating between high and low strains and with a few cycles. You can see using our gels, the viscosity is actually quite low, although it does recover with this multiple step strain format. And so we decided to add another modification, which is a pre-gel flow, which can be obtained from Rocket. It is a pre-gelatinized starch that is commonly used to thicken sauces, dressings, crunchiness to snacks, etc. But we decided to use it in a biomedical purpose. So after adding pre-gel flow, you can see that there's significant increase in gel viscosity, along with retention of the rapid viscosity change after multiple step strains experiments. And this rapid change is proof that the bio-ink could quickly return to its viscous entity after being extruded to retain structural integrity. This indicated that a suitable 3D printing bio-ink has been developed. Let's look at a video to illustrate this process. The first printhead is printhead 1, and it contains the gel and gum brief gel flow composite. A layer of more rigid precursor would first be deposited on the print bed. And then next, the cell containing more fluid collagen could be deposited into the precursor layer, after which the print bed can be heated at 37 degrees Celsius to cross-link the collagen. Then the gel and gum cross-linker can be added into the petri dish for the hydrogel to be fully fabricated. So again, doing rheological sweeps for amplitude and frequency, we show that our 3D printed hydrogels have similar rheological properties as our hand fabricated variants. You can see the overlap of all the curves. And for physical characterization, we look at the print resolution by printing the gel and gum pre-gel flow composite in filaments, where we then compare the average width of the filaments to the diameters of the nozzles. Due to minimal clumping, the hydrogel filaments are just 10% wider than the nozzle. Over here, diameter. So next we examine the shape fidelity factor, or SFF in short, of the 3D printed hydrogels. We compared the average surface area of the 3D printed films against the computer-aided design model. By dividing them, we noticed that the 3D printed hydrogel films are about 36% wider than the design model. Now, these parameters are all within acceptable published range. For the 3D printed stem cells hydrogels, MTS assays were performed, and we showed that the 3D printed gels were about 23%, showed a 23% decrease in cell viability when the cells were 3D printed, probably because of the stress of extrusion. And this is also in accordance with literature, most common literature. And from the confocal images, we can see that 3D printed stem cells were able to adhere, although there's a slight decrease in cell number over 14 days of culture. So this, in conclusion, I would like to end by saying that natural materials is really a more sustainable raw ingredient that can be mined for the ability to form hydrogels. We have already shown with our studies thus far that it is possible. It's very easily made and fabricated, and the formulation strategies rendered the nature-based hydrogel cell adhesive for the successful encapsulation of stem cells. So there is quite a lot of potential, and not just as alone, to use it as alone, but also to deliver stem cells. And finally, 3D printing of these hydrogels makes it amenable for personalized biomedical application. And this also shows that our raw materials are pretty good as bioinks for 3D printing. Finally, I would like to acknowledge my lab members, past and present, for their support and hard work. And Jian Yao over here is the main driver of this work. I also want to thank my funding sources to run our lab, without which I would not be here to share this exciting work with you. I thank you for listening.
All right, thank you to Professor Dr. Ooi. So we're going to continue with the next presentation. So the Q&A will be moved until the presentation of all three speakers. So we go with our next speaker for today, this afternoon. So our next speaker is Dr. Tan Wei. She's an assistant professor at UMass. So let me briefly introduce you guys to our second speaker. So Dr. Tan Wei received her bachelor's degree in chemistry from University of Science and Technology of China in 2011, and she pursued her PhD degree in chemistry from University of Georgia in 2016. After she received a postdoctoral training at NIH, she joined NUS as a professor, currently in the departments of pharmacy and downstream radiology in 2021. So her research interests are focusing on development of safe and effective drug-delivery technologies to empower early detection, precise stratification, and personalized therapy for cancer. So without further ado, we would like to invite Assistant Professor Tan Wei to deliver her presentation.
Hello, everyone. My name is Tan Wei. I'm Assistant Prof in Pharmacy at the National University of Singapore. It's my great pleasure to share my research here at the Malaysia Technology Expo 2022. My topic is sustainable delivery sustainable development of drug delivery nanosystems for tumor hypoxia modulation. First, I would like to briefly introduce myself. I got my bachelor degree in chemistry in China, and then I went to the United States for my PhD study. After my postdoc training at NIH, National Institutes of Health, I moved to Singapore to establish my own research lab in October 2021, so about 1 year ago. And our research mainly focuses on drug delivery and nanomedicine, and we aim to develop novel, safe, and effective drug delivery technologies to empower early detection, precise stratification, and personalized therapy for cancer, as well as other human healthcare challenges. And we integrate chemistry, biochemical science, biomaterials, nanomedicine, molecular imaging, as well as immunoengineering fundamentals to fulfill this goal and to study the relationships between in vivo drug transport and the biological barriers to the drug delivery systems. And we are currently establishing a new research lab. So we are currently actively recruit talented graduate students and research fellows to join us for exciting research in nanomedicine and drug delivery. And the nanomedicine, of course, I think everyone has to admit that it has played an important role in the prevention of COVID-19. And the two mRNA vaccines are all based on lipid nanoparticle formulations. They have achieved a tremendous success. In a typical mRNA vaccine, the lipid nanoparticle consisted of 4 lipid components: ionizable cation lipid, which help to bind and protect RNA from degradation and improve its binding and improve its encapsulation efficacy. And in the acidic tumor microenvironment or the acidic lysosome, it can turn into positive charge and facilitate the endosome escape of the mRNA therapeutics. And a third type of lipid components in LNP is the cholesterol, which is important for endocytosis of the nanosystems. And you also include phospholipid, hyperlipid, and the PEG-based lipid components for stabilization of the particles and protect the nanoparticle from opposition. In addition to that, we believe nanoparticle or nanotechnologies have great potential to improve the existing biomedical technologies and also enable entirely new strategies and applications in human healthcare. It induces lower costs and materials usage, therefore it is more sustainable comparing to conventional drugs or medicines. And there are a few nanomedicines that are already in clinical trials. This can be considered, therefore, the nanoparticle formulations can be considered as new drugs because of their new biodistribution and PK or PD profiles, which may yield new efficacy profile. And they can also be considered as new therapies because it can integrate new modalities, new targets, and also new dosing regimens for new treatment efficacy. And another important advantage is that we can always surface functionalization of the nanoparticles with targeting ligand for targeted drug delivery, which help them to overcome the biological barriers and achieve a higher accumulation at the disease sites. And also the nanoparticles, which functionalize of imaging functions, can be used for early disease detection and prediction and monitoring the treatment efficacy. We can also integrate these kind of nanoparticles or nanomaterials into diagnostic devices, which can achieve high sensitivity. And when we combine or integrate nanoparticles that have both diagnostic and therapeutic actions, we call it nanotheranostics or theranostics, which can achieve diagnostic and therapeutic performance at the same time. And there are already a few nanomedicine approved for cancer treatment. For example, the Abraxane, which is albumin-binded paclitaxel, and it is approved by FDA in 2005 for metastatic cancer or breast cancer. And there are a few liposome-based nanomedicine, for example, the doxorubicin, which approved in the early 1995 for the treatment of HIV-related diseases, and also later it has been used for the treatment of metastatic breast cancer as well as metastatic ovarian cancer. And we also have protein-based and the polymer nanoparticle-based nanomedicines. Moreover, there are a few more nanoformulations are currently in clinical trials or close to filing the investigational new drug applications with FDA. However, there are a few questions need to be answered, or there are a few roadblocks need to be cleared for the sustainable development of nanomedicine. For example, we need to understand how do nanomaterials interact with living cells and tissues, because the interactions between nanomaterials and the biological environment or biology, tumor biology, are actually complex and poorly understood. And a second question is always about the nanoparticles toxicity. What are the determinants of the nanoparticle toxicity? Does the size, surface chemistry, or the surface charge important factors regarding its toxicity, or will the purity of the materials a key player? And maybe is the nanoparticle must be fully biodegradable for safe use? A few questions or a series of questions need to be answered. And the third question, I think, should be the unclear or poorly understand or even highly variable nanoparticle fate in vivo. A few factors need to be taken into consideration regarding this question. For example, maybe the size, surface charge, again, and the surface coating dependent, and also the hydrophobicity of the nanomaterials, and the renal clearance, and the MPS system capture, or maybe also the delivery method, for example, intravenously injection or comparing to IP injection. Thus, these factors impact the final fate of the nanoparticles in vivo. And last but not least, there are some concerns in the batch-to-batch variability in nanoparticle manufacturing. This may further affect the nanoparticle toxicity and bring issues such as reproducibility and reliability of the nanoparticles performance. Well, in my research today, in my talk today, I will share some of my research on the development of nanosystems for tumor hypoxia modulation. So what is tumor hypoxia? How does it happen? And tumor hypoxia is the situation where the tumor cells have been deprived of oxygen. It developed mainly due to the uncontrollable proliferation of the cancer cells, altered metabolism, and abnormal tumor vessels. The cancer cells maintain a high respiration rate to fulfill the high energy demand. This leads to an imbalance between oxygen supply and the consumption, therefore resulting in hypoxic areas that may negatively impact the therapeutic outcomes. For example, the photodynamic therapy, PDT for short, or the radiation therapy, RT for short. In PDT, let's look at this chart here. The photosensitizers can be activated by laser light at specific wavelengths, transfer the light energy into the nearby oxygen in the tissues, and generate toxic free radicals or single oxygen, or in general, the reactive oxygen species for cell killing due to the cellular toxicity from reactive oxygen species. While in RT, the ionizing irradiation first induced the generation of free radical DNAs. In hypoxia, the oxygen plays an important role to fix the DNA damage and further induce the DNA break and finally the cellular death. Whereas in hypoxic conditions, the free DNA radicals may be restored or restitution into its normal state. Therefore, reverse the therapeutic efficacy of the radiation therapy. So according to both charts, we can clearly conclude that without sufficient oxygen supply, the therapies may be very inefficient. And therefore, we propose to use nanotechnology to site-specifically deliver oxygen to tumor sites and therefore overcoming hypoxia-associated resistance to anti-cancer therapies. So in my first study, I used the red blood cells, or RBC for short, which is a natural oxygen carrier for oxygen delivery. We developed a technique called RBC PDT. And during RBC PDT, RBC would continuously release oxygen, shown as blue bubbles here, the right figures, and generate an oxygen-rich zone or close to the RBC surface. The photosynthetizer attached to the cell surface, shown in white spheres there, then therefore can benefit from the oxygen flow to continuously produce single oxygen, even under hypoxia conditions. So the most challenging part in this study was how to load a large number of photosynthesizers onto the RBC surface, as most photosynthesizers are rather hydrophobic and not provide conjugation-friendly functional groups. To solve this problem, we enriched the photosynthesizers into a ferritin nanocage. It's a protein cage with hollow cavity. It is used for iron storage in humans or other living organisms. We then conjugate this nanocage onto the RBC surface while avidin biotin bridge. And in the right figures here, the SAM shown that in the modified RBC, they shown a coarse surface with many nanoparticles on it, and the loading efficiency is pretty high, which is determined to be about 20 million photosynthetizer molecules per RBC. And to examine whether RBC PDT can effectively produce single oxygen even under hypoxic conditions, we developed an acute hypoxic tumor model and applied PDT after intratumor injection of different photosynthetizer formulations. After that, we performed the tallow assay to evaluate the tumor apoptosis. we find similar level of positive fluorescent signal, which indicating the tumor cell apoptosis in oxygen-treated or non-treated RBC photosensitizer group. In contrast, the carbon monoxide pre-treated RBC photosensitizer group, which without oxygen carrying capacity almost lost their ability to induce PDT damage to tumors, and which suggested that the oxygen is a key player in this RBC PDT technique. And the RBC technique gained valuable insights on how to use a nature oxygen carrier to address the oxygen limitation in PDT treatment. However, the RBC although have a long circulation half-life and a minimal actualization outside of the tumor blood vessels, it also confined the PDT damage only to tumor blood vessels, but not directly to cancer cells because they cannot actualizate into the tumors, only circulate in the red blood cell circulation system. So after we noticed this limitation, in our second study, we developed a nano-sized artificial RBC delivery systems. We call it PFC PDT technology, in which we use the perfluorine carbon nanodroplet, or PFC for short, as a carrier for both oxygen and photosensitizers. The PFC has high oxygen capacity and is the main category of oxygen-carrying blood substitute in the clinic. Moreover, some perfluorine carbon nanodroplet has very low boiling point, so therefore they can easily be vaporized by thermal effects to release their carried oxygen. So in PFC PDT, we stabilized the PFC nanodroplet with a photoacoustic PDI molecule layers. With the laser irradiation, the local temperature can be increased due to the photo to thermal conversion with the PDI molecules. And the increased temperature will trigger the PFC vaporization to induce a strong ultrasound contrast, as well as lead the release of dissolved oxygen in the PFC nanodroplet. Meanwhile, the photosensitizer encapsulated or dissolved in the PFC nanodroplet can be activated by the same laser and for oxygen-promoted PDT effect. And we performed the tallow assays to evaluate the cell apoptosis after different treatment, which is the PFC PDT and the conventional PDT without further oxygen supply, but only the photosensitizer and the PDI self-assembled nanoparticles. And we investigate the PDT effect in a dose-dependent manner, and we, according to this tallow assay result, we find that the PFC PDT with PFC nanodroplet, which can carry external oxygen molecules, showed a better efficacy than conventional PDT, while the PFC PDT, while the conventional PDT showed a much less efficient therapeutic efficacy comparing to PFC PDT under the same irradiation dosage. And this result suggested that PFC PDT can effectively deliver oxygen to tumors and promote PDT efficacy. And after these two studies, we start to think whether we can further boost or increase the tumor oxygenation level by developing a more efficient oxygen delivery systems. When we revisit the origination of tumor hypoxia, as I introduced in my previous slides, we find that in addition to improving external oxygen delivery or oxygen source, the unique physiological feature of the high cellular oxygen consumption might provide us an opportunity if we can find a solution or nano delivery systems to suppress cancer cell respiration, in addition to the external oxygen delivery, the physiological oxygen can be conserved, while the delivered oxygen can be fully used for anti-cancer therapy. And so in this study, we propose to deliver an NO nitric oxygen prodrug for the reduced oxygen consumption via external oxygen delivery for increased oxygen supply. Design delivery systems in the PFC PDT study, we immobilized the PFC perfluorine carbon chains and photothermostic semi-conducting polymer brushes into the framework of silicon nanoparticles. And this immobilization of perfluorine carbon chains avoid the easy to vaporization characteristics of the free PFC nanodroplet, therefore decreasing the difficulties in temperature control for the oxygen loading and the storage. While the semi-conducting polymer brushes here can be used as a photothermal controlled can be used for photothermally controlled oxygen release. And in the meantime, we in situ hybridized or polymerized hydrophobic alkyl chains onto the inner shell of the nanoparticles for enhanced loading and retention of hydrophobic NO nitric oxide prodrugs, which can release NO in response to the acidic tumor microenvironment. And we investigate the in vitro NO and oxygen release by using fluorescent indicators after different treatments in hypoxic cells. According to the confocal imaging results, we find that the oxygen release was dependent on laser irradiation, but the NO release was independent on that. And interestingly, we find that the NO release elevated the hypoxia to some extent by showing a decreased red fluorescence signals, and this was because the NO can competitively bind to the oxygen binding site in the cell respiration chain in mitochondria, therefore reducing the cellular oxygen consumption. And similar results can be observed in vivo. Based on the photoacoustic imaging on tumor oxygenation level, we can conclude or we can find that the nanosystems could relieve the tumor hypoxia upon its gradual accumulation at the tumor sites due to the gradually released NO in response to the acidic tumor microenvironment, while a subsequent laser irradiation can further enhance the intratumoral oxygenation level, which attributed to the laser activatable oxygen release. And the increased tumor oxygenation resulted in boosted RT therapy, as shown in the tumor growth curves here. The X-ray irradiation alone, shown in the pink curve here, only showed a moderate tumor inhibition rate of around 50%, while the rate increased to 70% in the oxygen econometer boosted RT therapy shown in the blue curves here. And more excitingly, the photothermal effect can be further increased to induce a higher temperature at the tumor site, therefore eliminate the tumor residues with radio-resistant properties due to the factors other than hypoxia. Taken together by combining photothermal therapy and oxygen-boosted radiation therapy, shown in red curves here, we achieved complete tumor regression. So all these studies demonstrated that by modulating tumor hypoxia with nano drug delivery systems, we can significantly overcome the hypoxia-induced resistance to some the tumor therapies. And in summary, the nanotechnologies has great potential to improve the existing biomedical technology and enable entirely new strategies and applications in healthcare. And the tumor microenvironment plays an important role in determining the therapeutic outcomes of nanomedicines. And last but not least, by modulating the hypoxic tumor microenvironment with an on-demand oxygen delivery technology, for example, RBC-PDT, PFC-PDT, and oxygen economizer boosted radiation therapy, we have successfully overcome the tumor hypoxia-related resistance to anti-cancer therapies. So that's all for my presentation today. Thanks very much for your attention.
Thank you so much for the presentation to Dr. Tang Wei. So we shall proceed with our next presenters. So our next presenter will be by Assistant Professor Dr. Chen Wen-Chen. So let me introduce the background of our speaker. So Dr. Chen Wen-Chen, or Dr. Chen, has intensive experience in the downstream processing of proteins and also peptides through membrane filtration and crystallization. Over the past 10 years, Dr. Chen has collaborated closely with global pharmaceutical companies, including GSK, Pfizer, Lonza, and Fujifilm Diosynth in developing novel downstream processing technologies at Imperial College General ICL in UK, where also his alma mater, where he obtained his undergraduate and PhD degree in chemical engineering. So for today, Dr. Chen will explain his topic in membrane cascade design for sustainable pharmaceutical processes. So without further ado, I would like to invite Dr. Chen to deliver his talk. Please welcome.
Hello, everyone. Many thanks for coming to my talk today. My name is Wen. Today, I will talk to you about the design of membrane cascade systems for sustainable pharmaceutical processes. Hopefully, by the end of today's talk, you will understand why membrane filtration is useful in the pharmaceutical context compared to other separation processes, and how membrane cascade design in particular can be useful for achieving sustainability. So now let's look at my profile before we move on. My name is Wen. I am an assistant professor at the Department of Pharmacy at the National University of Singapore, NUS. My research interest mainly lies in membrane filtration, crystallization, and protein peptide purification and formulation in general. Here is the link to my LinkedIn profile. If you're interested, you can also scan the QR code on the bottom right corner to follow me on LinkedIn. So now let's talk about separation science. Why is it so important? In the industrial context, we many times come to the point where we will have a mixture of useful compounds and impurities. If you look at the right, we have a figure here. The orange cross represents the target compound that we are interested in. It comes with two impurities, which is the blue circle and the green triangle. Our goal is to separate the target molecule from the impurities. This can be done using different separation techniques. And at the end, we will have the target compound in high purity. And then for the impurities, we will remove them. And it is possible to further purify them in such a way that we can sell them separately to people who are interested in their applications. So in short, separation science is crucial in the industrial context for delivering pure target compounds. There are many separation techniques that people have used, including distillation, absorption, extraction, precipitation, crystallization, and then lastly, membrane filtration. Let's go through them one by one. For distillation, it is heavily used in the oil and gas industry, where the liquid is actually heated, and distillation make use of the difference in volatility of the compounds. By doing so, the compounds can be separated very, very precisely, but distillation can be highly energy intensive and may not be suitable for compounds that are heat sensitive. Now, let's move on to absorption. In the industrial context, we use a lot of something called absorbent to remove target compound or impurities. This absorbent comes in the form of a solid, and they can interact with either the target compound or the impurities via different mechanisms, including affinity binding and charge interactions. In the industrial context, many people use a process called chromatography, where a solid packing material will be packed into a column, and the liquid that contains the compound of interest and the impurities will be passed through the column to interact with the packing material. The advantages of chromatography is that it can be very precise and to give very high purity and high yield of the compound, but it can be very, very difficult to scale up beyond the ton scale. This is because it is quite challenging to pack large columns and to pass through the column with a large amount of processed liquids. Next, we move on to extraction. This is where we use usually two liquids that are not miscible, and they have different affinity degree to the target compound or the impurities. At the end of the process, you will have one of the liquids with high concentration of the target compound, and the other liquid in high concentration of the impurities. The downside for liquid extraction is that it could use a lot of solvent in the process, and that is not very sustainable. Next, we have precipitation and crystallization. In these two processes, either the target compound or the impurities are solidified using different mechanisms, such as cooling, or by adding anti-solvents or using additives. The good thing about precipitation and crystallization is that it is very easy to process the solids after it is formed, because it can be filtered off very quickly, very easily from the remaining liquids. The downside is that, for example, for precipitation, sometimes it may not be suitable for biomolecules, which are very easily denatured. And for crystallization, in the context of biomolecule, it can be quite challenging to crystallize large molecules such as protein. And then lastly, we come to membrane filtration, which is the focus of today's talk. Membrane filtration is advantageous as in number one, it is not energy intensive compared to distillation because it doesn't involve the heating and cooling of processed liquids. Number two, it doesn't involve the use of huge amount of absorbent, like in the case of chromatography / absorption. And this can bring down the cost of the process significantly. Next, compared to liquid extraction, membrane filtration often use much less processed solvents. to achieve the same separation. And then lastly, membrane filtration can very easily couple with precipitation and crystallization. And this shows the flexibility of membrane filtration working with other separation processes to achieve the separation performance that we need. Now, let's look at membrane filtration in detail. What is a membrane? It is essentially a physical barrier that the processed liquid or gas has to pass through to achieve the separation of compounds. There are many membranes available in the market, and I would like to use the asymmetric membrane as an example for today. You can look at the right-hand side of this slide, and here you can see that for the asymmetric membrane, it has an active layer on top for the separation and a porous support at the bottom that gives the mechanical strength to the material and contribute to the control of the flow rate that passes through the membrane. In this case, for example, we have a mixture of A and B, and they are different in size. By choosing a membrane that has an active layer that has a pore size larger than the size of B, but smaller than the size of A, we can allow B to pass through selectively while retaining A on the other side. There are many types of membrane filtration, and they are characterized based on the particle size that they can separate. Now let's look at the first one, which is microfiltration, or MF in short. It is useful for separating particles or molecules with size between 0.1 to 10 micron. Next, we have ultrafiltration, UF, which is useful to separate compounds or particles between 2 to 100 nanometers in size. Next, we have nanofiltration, NF, which is useful to separate compound or particles with size less than 2 nanometers. Last but not least, we have reverse osmosis, RO, which is useful to separate solvent, and in many times it is water, as in the context of distillation. This is a typical membrane filtration system, and it starts off with the membrane unit, and then we will have a feed tank to supply the processed liquid to the system via the pump here. And the pump usually generates heat during the process, and that's why we usually have a temperature cooling system here, so that the liquid, the processed liquid, can be maintained at the same temperature. Pressure is needed for membrane filtration processes, and it can be supplied by gas, as in this case by nitrogen gas, or by a pump that gives the pressure. Whatever that comes through the membrane unit goes into something called a permeate tank. It can contain the compound of interest or the impurities that we want to remove. So now let's take a closer look at the membrane filtration unit itself. In the unit, we have the membrane separating two compartments. The one that contains the liquid that passes through the membrane is called a permeate compartment, and it comes from the word permeation. And on the other side, we have something called retentate compartment, and retentate comes from the word retain. This is where the feed comes in through the inlet, and part of the feed under pressure goes through the membrane into the permeate compartment, and the remaining is called a retentate. And this goes through the outlets on this side and being recycled back to the system. If we look at it closer, you will realize that the flow rate of the feed stream must be larger than the flow rate of the retentate stream, because part of the feed stream goes through the membrane. And this is what we call the transmembrane flow rate, as we show here. And one important concept I want to introduce to you is called rejection. So before I explain this mathematical definition, rejection of a compound, when it is zero, it means everything passes through the membrane. That means all of your compound of interest pass through the membrane without being retained. And when rejection is 100%, the compound of interest is being retained completely by the membrane. And let's see how we calculate this rejection. And this is, we will first need to know the ratio of the concentration of the compound of interest in the different compartments. So we need to look at the concentration in the permeate compartment over the concentration in the retentate compartment. When the permeate concentration and the retentate concentration for the compound of interest is equal, this means the compound passes through the membrane completely without being retained at all. However, if the concentration of the compound of interest in the permeate stream is zero, you can see that this ratio also becomes zero. And because of that, 1 minus 0 is 1, and that's why the rejection is 1 or 100%. This rejection concept is very, very important for our talk later on. So please pay attention. So now we come to the topic of today's talk after all the background information. So what is a membrane cascade and why it is needed? On the right-hand side, you can see that we start off with a single membrane system like I showed you just now. It consists of a feed tank, mainly a feed tank, and a membrane unit. However, what we found out is that, especially in the case where we would like to retain the compound of interest within the system, even though with the system, and what we do is usually we add fresh solvent into the system to wash out all the impurities through the membrane while retaining the product within the system. What we found out is that this can be quite problematic when we, even when the rejection of the compound of interest is very high. So we have a concept called wash volume. This is equal to the amount of fresh solvent that we add into the system normalized by the total volume of the system. So the more fresh solvent that we add in, the higher the wash volume. And what you can see is that even with a 90% rejection of the compound of interest, by doing a 10 wash volume washing, we can lose up to 60% of the yield through the membrane. This is where the membrane cascade is useful. There are two things that the membrane cascade can do. Number one, it can increase the yield of the recovery significantly for the compound of interest. And secondly, it can also perform separation of multiple compounds at the same time. So before we move on to the case studies to explain the use of membrane cascade, we would like to just recap what we have learned so far. Number one, there are many separation processes for separating the target compound from impurities. And membrane filtration is, in particular, very, very sustainable because, number one, it is less energy intensive. Number two, it uses a lot less solvents for washing or for processing. And number three, it can couple very easily with other separation processes, giving it very high flexibility. And because of that, membrane filtration is useful in sustainable process development. And the importance of membrane cascade is to, number one, achieve high yield of recovery for the target compound. And number two, the possibility of performing multiple separation at the same time. I would like to use a case study to demonstrate the usefulness of membrane cascade for achieving sustainability. In this case, we would like to perform liquid phase peptide synthesis in a membrane cascade system, where the peptide that we are looking at are basically shorter versions of proteins. They are peptides, they are polymers of amino acids with less than 50 amino acids linked covalently together. The conventional method that people use for peptide synthesis is called solid phase peptide synthesis, or SPPS in short. In this case, the peptide is synthesized on insoluble resin by the addition of amino acid one by one onto the resin. The advantage of this method, SPPS, is that it is very easy to separate the peptide attached onto the resin from all the reagents that are used. And that's why the peptide synthesis method can be very simple for SPPS. The challenge for SPPS is that it is very difficult to scale up beyond the kilogram scale because of the challenges in dealing with solids. So the method that we develop is based on liquid phase peptide synthesis, where a soluble anchor is used for the peptide synthesis using similar addition of amino acid one by one. And this soluble anchor can help the compound to precipitate very easily for washing and further processing. For the method that we use that uses membrane system is called membrane enhanced peptide synthesis, or MEPS in short. Essentially, we replace the precipitation step with membrane filtration. And by doing so, we can achieve sustainability, as I will show you in a case study later. So in short, this is how we perform the process. We start off with the anchor, the soluble anchor, that cannot pass through the membrane. We first attach amino acid onto this anchor. This amino acid is protected by the FMOC group. So the resulting material is the FMOC protected amino acid 1, here abbreviated as AA1, attached to the anchor. After this, we perform our first membrane filtration step to remove all the reagent used in the coupling step. And then we move on to the deprotection stage, where we remove the FMOC group in such a way that the next amino acid can be attached to the first one. And after this deprotection step, we will do the membrane filtration to remove all the reagent, and then we will repeat the coupling and the post-coupling filtration, and we will repeat this cycle n-1 times, so that at the end we will achieve this, we will obtain this FMOC protected peptide on the anchor. And to finally get the material, we will remove this FMOC group and also remove the peptide from the anchor. So now let's look at some numbers. For 6 amino acid peptides, we perform the MEPS, where the anchor rejection is very high, 99.7%, whereas the rejection for all the excess reagents are very, very low, about 33.0%. To look at the sustainability of the sustainable nature of the process, we use the E factor, which is very commonly used in green chemistry. It is mathematically defined as the total mass of waste generated by the process divided by the total mass of the product. We synthesize these 6 amino acid peptides using the solid phase peptide synthesis method, the liquid phase peptide synthesis method, and also the membrane enhanced peptide synthesis method, MEPS. You can see that in terms of purity and yield, the MEPS is very comparable to the SPPS and LPPS, but the E factor is significantly lower. This shows very clearly that the MEPS is much more sustainable compared to SPPS and LPPS. However, one challenge of MEPS is that when it is conducted in a single-stage system, it will result in significant loss in yield. So you see at the end, the yield is only about 71%, and it decreases over time as shown here. This is where the membrane cascade becomes useful. By installing an additional membrane unit right after the first one, this allows the recovery of the useful peptide on the anchor that is lost through the membrane back to the main system. And by doing so, you can see that the yield can be consistently maintained at more than 90%. And this is crucial for driving the E factor down further and making this membrane process, peptide synthesis, a lot more sustainable compared to the rest. To conclude, many thanks for your attention today. We know that membrane filtration is a sustainable separation process because it has low energy intensity, low solvent usage, and high precision separation based on molecular size, charge, and hydrophobicity. The key considerations for membrane performance are rejection and permeate flux. In particular, membrane cascade is a very powerful system that can enhance the recovery yield of the compound of interest. And it can also allow the separation of multi-components at the same time. And it can improve the E factor, which is the sustainability of the membrane process. Many thanks for your attention. If you have any question, please do email me at wen.chan@nus.edu.sg. Once again, if you want to connect me via LinkedIn, this is the QR code. Many thanks for your attention today, and hope that we will work together very soon to achieve sustainability for the world. See you.
Okay, thank you so much to Dr. Wen Chen for the presentation. So we are now entering the Q&A session. So as you can see, we have all the 3 presentation from the natural-based materials, later to the nanomaterials, and after that, into of using a membrane to part of the sustainable pharmaceutical process. So from material up to process, that's what we try to deliver today. Okay, so for the Q&A session, as I mentioned earlier, so you may use the Q&A function at the bottom of your Zoom, or if you want to have a direct question, please raise your hand and I will call you upon to ask the question directly to our speakers. So with us today, we have Dr. Tang Wei and also Dr. Wen Chen online with us. So how are you, Dr. Chen?
Hello. Yes, I'm very good. Many thanks for being the MC for today. Yeah, I think you have done an excellent job. Thank you.
Oh, thank you. So yeah, you're in Singapore. I'm pretty sure you're in Singapore right now, right?
Yeah, I'm in Singapore.
So directly from the campus, or you're working from anywhere at present?
Yeah, I'm at the campus.
At the campus. Okay. So so we have we were waiting if there is a question from the members. So like, I quite interesting because when you have presentation, when you compare with conventional method of the solid phase peptide synthesis or SPSS, right? Because you mentioned that because like it can goes up to upscaling, but when it comes to more than kilogram amount and over, because like there are a lot of waste production and so forth. So it's a bit hard to have the high purity of the peptide. So with your method, how was it that can be, and the one that you just shared with us just now, like how was it that can ensure the higher yield and purity interval of upscaling kilogram and more?
Yep. Many thanks for the question. So just to before I move on to talk about the new technology. So for the solid phase, it's actually a fantastic technology. But the only problem is that when you try to scale it up, doing the mixing, okay, in the presence of the solid resin is always problematic because the resin, if you stir it for too hard or too fast, it can break. Okay, and this will be problematic for the method. And what we are doing is actually essentially liquid phase peptide synthesis attached onto this what we call a huge anchor, which can be retained by the membrane. So by doing so, actually, it is a very how will I call it, a very scalable liquid phase process, and it also overcomes some of the problems in solid phase, right? Because especially if you grow longer peptides, it can actually interact with the polymer matrix, okay, with the resin. But in the liquid phase method, it's not such a problem.
Okay, so yeah, I mean that's because like usually like because for me myself, I'm working in nanomaterials. Like peptide is one of the material that we always have difficulties of scaling up. Actually, when you synthesize it on peptide, like because we try the solid phase, we try the liquid phase, and so on. Like as mentioned right now, like perhaps like with the one that you share, like is it also feasible to be done in the lab scale or just like appropriate for industrial scale only?
I did at the lab scale. I think that the scale was about a few hundred milliliters. Okay. The only thing is that that the anchor compound is something that may not be readily available in the markets. It's something that we synthesized in the past, but otherwise it's very scalable. You can do it from just tens of milliliters all the way up to liters. It's doable.
All right. So I think that's one of the approach. Like I think like it's also come a thing after the COVID hit. Like people start with composition modeling first because usually before we would just like try and error doing all of the synthesize. Right now we start with composition modeling and afterwards it goes to with the actual experimental work. So do you think with that approach will give a better yield and cut the whole process? I mean, like we talk about sustainable, like rather than you waste a lot of things, try and error. Composition start first and then do synthesis. Would that be better from your own opinion?
Yes, I think for the modeling, I think it also depends on the maturity of the technology, right? Sometimes if you're dealing with new technology, having the right data to support the right model can be problematic. So for example, for my work, I did the experiment first, and then I built a process model for it. But I have to say, to really make sure that we can have a satisfactory model, we need more data.
That's true. Because like I mean, usually composition you have already some kind of data that people done before. That's why you kind of have improvement. But like for example, like in your case, you're totally making a new peptide. I mean, you are the one who create the data, right? So without the data, it's hard for you to do the modeling afterwards.
Yep. So that's why I think if we have, for example, a big enough data set, so let's say if I keep making a peptide up to 10 amino acids, and I have different yield and purity, and I can use that data set to estimate.
Yeah, that's true. Like, okay, thank you, Lucian. So like, hello, Dr. Tang, can you hear me? I think you are on.
Hello, yes.
Okay, yeah.
Hello.
Yeah, I like your background. I missed that ground at NUS.
Oh.
I missed that one. Like when I was there, like I feel like, am I in Singapore? I mean, in Europe, it's kind of like that kind of vibe that NUS managed to develop that ground, right? Yeah.
True.
So,
yes.
So you also live from the campus, or you're somehow from the cafe? Kind of find people working from anywhere right now.
Campus.
From campus. Okay.
Yeah.
Okay. So yeah, I like your presentation because like you talk about using PFC, right, as part of your nano carriers to deliver the oxygen. Like, because I never work with PFC because usually with one material, nanomaterial that I work by based on my experience, I work with other polymeric base or organic like carbon or graphene. Like, So why exactly PFC been chosen, like to deliver O2? Like, what make is the most suitable to deliver oxygen in your case?
So for the PFC, we choose it because it itself has a high oxygen loading capacity. In the clinic, the PFC has been used as a oxygen carrying blood substitution. So it's an excellent source for oxygen delivery. But I believe the challenging is to make it nano size, and some of the PFC components is very unstable. They prefer to convert to gas phase rather than stay in liquid phase. So we use nanotechnology to stabilize the PFC nano droplet.
Okay, yeah, because that's also I would like to ask this you answer, because like PFC known for the instability, right?
Right.
So,
yes.
So like, if you don't mind, I don't know whether if the data is PNC or not. Like, at what range of size do you manage to get for your PFC?
You mean the PFC nano droplet?
Yeah.
It's around 100 nanometer.
Oh yeah, that's mean it's just a nice size for delivery to the tumor, like 100 to 200. That's the normal size people do. So okay, like, because you say it's in clinical people use that as for the blood oxygen carrier, right? So like, how exactly when you change that into a nano droplet, like it allows it to release the oxygen rather than just conjugated or strongly bonded to the PFC, the oxygen itself?
The oxygen itself I believe it's the certain interactions between the oxygen molecule with the carbon fluoride bond, right? So in our follow-up study, we immobilize the carbon fluoride bond, the carbon fluoride chains into a hybrid nanoparticle, and by doing this, we can avoid the avoid the instability issue and to make it stable within a hybrid nanoparticle.
Okay. Yeah, because like not many actually doing. I mean, I never heard that much people talk about delivering oxygen. Usually, people talk about when talk about cancer, you're delivering the fluorescein, you're delivering the anti-chemotherapy drugs or certain DNA or protein. But like, hearing your talk today, like, oh, people do also deliver oxygen using a nanotech, which interesting to explore further ahead. So, like, for example, like what do you, for your own opinion, like other than PFC, is there any other materials that are also suitable to have a same function like what you're doing right now?
Yeah. So for the drug delivery, we can use red blood cells, which I should have introduced that in my first example.
You modified the RBCs.
We didn't modify that, but we because the hemoglobin itself can carry oxygen, right? So we saturate red blood cells with oxygen, and then during the circulation, the oxygen will gradually release from the RBC red blood cell carrier. While other strategies for oxygen delivery may not directly deliver oxygen molecules themselves, they may deliver some oxygen generation nanomaterials to the tumor, and then the materials interact with the tumor microenvironment. For example, like manganese dioxide interact with hydrogen peroxide to release oxygen, to generate oxygen. Yeah, some redox reactions happen at the tumor site.
Okay, yeah. Kind of interesting because like because looking like you're using PFC modifying to nano size and have oxygen, like people are thinking we don't have actual replacement for blood right now, right? I was thinking like, is there any potential to like modify this one to have artificial blood in the future? Do you think is it possible?
Yeah. Yes, actually it's used in the clinic already.
Yeah.
For the
like,
as the blood substitutes.
For those countries have limitation on certain blood group, maybe like, I mean, working together with Dr. Winsha and have peptide work over there, like kind of like maybe we can produce like the world first approved synthetic blood soon. Who knows, right?
Looking forward that.
Yeah, because like it will help throughout the world of the blood supply, and even for me who's scared of needles to donate my own blood. So I think that also will help a lot of people to like you have that a supply artistic blood group, and then with protein work, and since a lot of protein work, that's mimicking how the blood works, with your material also like delivering oxygen. I think that kind of like can save a lot of people. Yeah,
hope so.
Yeah, I hope so. Like, because like people talk about we don't have any artificial blood yet. We don't have artificial blood. Like, who knows that with collaborating with a lot of people from expertise that we can develop one. Yeah, right. It's not impossible right now with sciences. Anything is possible. It's just a matter like at the actual time that we manage to get it the correct. I mean, like the eureka moment. Like, okay, now we make it.
Yeah, cool.
Yeah.
And
Okay.
we're looking for more collaborations.
Yeah,
and to
of course. I'm also like looking for
translate.
collaboration. Like, actually, like I'm missing to go back to Singapore now. Now, like things are open, the borders opening back. Like I'm missing to go back to NUS. Actually, like the last time we somehow connected with few members, but then couldn't manage after because the COVID hit, not just us, throughout the world globally. So I think that to be the next opportunity for collaboration between Malaysia, Singapore, and other part of the world too.
Yeah.
Right. Okay. I think we already like a 4:45, and that also marks the end of our Q&A session for today. And thank you so much again for Dr. Tang and Dr. Wen-Chen for your sharing. It's really a remarkable shares. Like I learned a lot by myself. Like even I work in nanomaterials, like I learned a lot from both of you and from other speakers as well. So thanks so much for your time, contribute your and sharing your expertise with all members in the Malaysian Technology Expo 2022 in Malaysia. So for all the members on the floor, thank you again for joining us for today. And don't forget the whole MT22 Malaysia Technology Expo SDG International Innovation Awards and Expo today is just the third day. So we have more few days until 21st October 2022. Please keep joining us for the next events throughout the other few days ahead. There will be a lot of things sharing for all the members, especially throughout the world with all the members here. So please stay tuned for further announcement for the next speakers, for the next event that are going to be organized for MT22. So on behalf of the committee for today's international webinars, thanks so much for everyone for joining us today. And then if you have any questions from NUS for the postgraduate program, or you're looking for collaboration, feel free to contact with all the members. You can go to website NUS, you can go to get contact with all our speakers today. Perhaps we'll get contact with one of them because that is so I can see a lot of opportunities to explore further ahead. So thank you again, everyone. I shall see everyone in the next program, and maybe not this time, maybe next year again. And please don't forget to follow all the information at website www.mte.org.my. For any updates on NUS, you can go to the website and also in the social media too. So thank you much again. Assalamualaikum warahmatullahi wabarakatuh, and have a great Tuesday evening, everyone.
Thank you. Bye.
Thank you, everyone. Bye.
Goodbye.